Hello friends, good morning to all of you. Today I am here with cell cycle and cell division part 1 of 11th standard. In today's session, we are going to discuss about cell cycle, basic processes in cell reproduction, phases of cell cycle and significance of mitosis. So let us start the session with cell cycle. Cell cycle was first of all described by Howard and Pele in 1953. Cell cycle is the sequence of events by which a cell duplicates its genome and synthesizes other cell contents and eventually divides into two daughter cells. Duration of cell cycle varies. A typical eukaryotic cell divides once in approx every 24 hours. Bacterial cell divides every 20 minutes. And yeast cell takes 90 minutes for division. Interphase of cell cycle lasts for more than 95% of the duration of cell cycle. Here it is the diagrammatic view of different phases of cell cycle. It consists of interphase and M phase, also called as mitosis. Interphase is divided into G1, S and G2 phases. And M phase is divided into prophase, metaphase, anaphase and telophase. The given figure also indicates about the cell life cycle. Now let us discuss about the basic processes in cell reproduction. First is about cell growth. Cell growth is the period of synthesis of and duplication of various components of the cell. Next process is cell division. When the cell matures, it divides into two daughter cells. As we have discussed earlier, interphase consists of G1 phase, that is gap 1, S phase, that is synthesis phase, and G2 phase, that is gap 2. M phase is also called as mitosis phase. Now let us move towards the phases of cell cycle. First is about interphase. Interphase means in between. Interphase involves a series of changes that a newly formed cells and its nucleus undergoes changes to become fit for division. Interphase is the period between end of one cell division to the beginning of next cell division. Interphase is also called as resting phase, but metabolically most active phase. It is the biosynthesis or energy phase. During this phase, cell prepares for next division phase. Interphase lasts for more than 90% of the duration of cell cycle, as we have discussed earlier, and it is further divided into three periods. First, gap 1, G1 phase, synthesis or S phase, and second gap or G2 phase. Duration of these phases varies in different organisms. Here you can see the three periods of interphase, that is G1 phase, S phase and G2 phase. Now we will study in detail about the events which are occurring in these phases. First is about G1 phase or gap 1. G1 phase is also called as post-mitotic or pre-synthetic or first growth phase. Here there is initiation of DNA replication takes place, but DNA do not replicate in this phase, only initiation takes place. The cell is metabolically active and there is synthesis of RNA, carbohydrate, lipids, proteins, including some non-histone proteins also. The duration of G1 phase is highly variable. During this phase, the cell grows double its original size and different organelles are formed. There is increase in the volume of cytoplasm in this phase. Another phase is S phase called synthesis phase. In this phase, there is stage of replication of each chromosome by synthesis of a new DNA molecule on the template of the existing DNA. Thus, DNA replicates and chromatids joined at centromere where, and this is called as primary constrictions. Here, as we have seen, that amount of DNA doubles per cell, but chromosome number remains same. In case of animal cell, centriole also duplicates in the cytoplasm. There is synthesis of basic proteins. The S phase is in most eukaryotic cells last for 6 to 8 hours. Now we will discuss about the G2 phase or gap 2 phase. It is a stage of further growth of the cell and preparation for its division. There is synthesis of spindle protein, types of RNA molecules, and synthesis of ATP and storage also takes place. Mitochondria and plastids duplicates here. If DNA is damaged during replication, then it get repaired in this phase. This is very important. G2 phase in most cell lasts for 2 to 5 hours. Now there is G0 or gap 0 or an extended G1 also called as quiescent stage. The cells which are not able to divide further and do not proceed beyond the G1 phase and start undergoing differentiation into a specific type of cells. Such cells are said to be in quiescent or G0 phase of cell cycle. Cells in G0 phase remains metabolically active but no longer proliferate under called on to do so depending on the requirement of the organisms. Some of the G0 phase cells such as fibroblasts help in healing of wounds, grow and divide again on the demand of the body. Now we will move towards M phase or mitotic phase. It results in separation of organelles, sister chromatids and their redistribution into daughter cells. It is called equational division because at the end of chromosome number remains same. M phase is divided into 4 stages of nuclear division. They are prophase, metaphase, anaphase and telophase. We will discuss here one by one. First stage of M phase is prophase. In this stage, nucleus of the cell becomes spheroid and there is increase in the viscosity of cytoplasm. The chromosomes shorten, thicken and become stenable at the end of prophase. It is due to condensation of chromosomal material. So how the chromosome is thickened? So that is due to condensation of chromosomal material. By the end of prophase, some chromosomes may contract up to 1/25th of their length in early prophase. In case of animal cell, centrioles start moving towards opposite poles. There is disappearance of Golgi bodies, endoplasmic reticulum, nucleolus and nuclear envelope at the end of prophase. And there is appearance of spindle apparatus in prophase also. In the given figure, you may observe the early and late prophase. You can observe the condensation of chromosome, in the late prophase and appearance of spindle apparatus as well as movement of centrioles to the opposite pole. This figure also depicts the early and late prophase. In the given figure, it is about G2 of interphase, prophase and pro-metaphase. Next is about metaphase. Metaphase is short and simple. There is complete disappearance of nuclear membrane, so no differentiation between nucleoplasm and cytoplasm. The chromosomes at this stage get attached to the spindle of their centromeres. Actually, spindle fibers attach to kinetocores, which are small disc-shaped structure at the surface of centromere of the chromosomes. In metaphase, there is maximum condensation of chromosome and it is the best stage to study the karyotype of chromosome, that is arrangement of chromosomes. Metaphase chromosomes clearly shows two sister chromatids held by centromere. All the chromosomes are usually lined in one plane to form equatorial plate, also called as metaphase plate. Occasionally, only the centromere lies towards the equatorial plane, while the chromosome's arms are directly away from the equator. In the given figure, you may observe how metaphase chromosomes are at equatorial plate in a line. Next stage is anaphase. The centromere of chromosomes divide simultaneously as anaphase starts, and the two chromatids of each pair separate and are now called daughter chromatids. These daughter chromatids move towards the two opposite poles and now they are called as daughter chromosomes. At this stage, these daughter chromosomes appear the shape of V, L, J and I depending on the position of centromere and kinetocores. In the given figure, you may observe the anaphase where you can see that daughter cells move towards the opposite pole. Now the next stage is telophase. Telophase is long and complex like prophase, and it is also called as reverse of prophase. Here there is restoration of interphase condition. There is decondensation of chromosome. Two sets of daughter chromosomes already reach to the opposite poles of the cell and there is disappearance of spindle fibers and appearance of nuclear envelope, nucleolus, Golgi bodies, endoplasmic reticulum. At the end of telophase, two daughter cells are formed. In this figure, you may observe the telophase. Now there is about cytokinesis. We have seen that nuclear material is already divided into two daughter cells. Now it is the time for division of cytoplasm. In animal cell, a cleavage furrow appears at the beginning of telophase, which becomes progressively deeper to form two daughter cells. But in plant cell, two daughter cells are formed by cell plate formation. During cytokinesis, there is distribution of organelles like mitochondria, plastids, etc. So here two daughter cells are formed by equal distribution of nuclear material as well as different organelles of the cell also. In this figure, you may compare the cytokinesis in plant cell and animal cell. This figure also shows about the cell's cycle and mitosis starting from that G2 of interphase to telophase and cytokinesis. Now the last topic of today's session is about significance of mitosis. Mitosis is restricted to diploid cells only, but in some haploid organism it also takes place. In mitosis, there is equal distribution of chromosomes, so it results in constant number of chromosomes. So that is why it is called as equational division. Why it is called as equational division? Because in the two daughter cells, equal number of chromosomes is distributed. It helps in repairing of cells of the upper layer of epidermis, cells of lining of the gut, and also blood cells. The apical and lateral cambium results in a continuous growth of plant throughout the life due to mitosis only. In multicellular organisms, growth of cell takes place due to mitosis. Mitosis is also essential for restoring nucleocytoplasmic ratio. So today this is the end of the session. Thank you. For any kind of doubts, please write your question in comment section.